Welcome back to our third installment for the digestive system. We have finally made it to the small intestine. Let's now address the gross anatomy followed by the histology of the small intestine. The small intestine is also called the small bowel. It's a coiled, thin-walled tube about 6 meters or 20 feet in length in the dead person. It is actually much shorter when you're alive due to muscle tone. We don't really know the exact length in the living human because it would be physically difficult to measure the intestine and would cause severe discomfort to the living individual anyway. The small intestine extends from the pylorus of the stomach to the cecum of the large intestine, and it occupies a significant portion of the abdominal cavity. The small intestine receives its blood supply, as we learned from Lab Unit 3, from the superior mesenteric artery primarily. And it consists of 3 segments, the duodenum, jejunum, and ileum. And I remember this as Down-Jones industrial. The kinds of movements that are capable by the small intestine are peristalsis and segmentation, as your book says, but pendular motions are also possible. This is the site where we are going to have the most chemical digestion and absorption. I will go through all 3 of the sections of the small intestine next. The duodenum, or duodenum, however you want to pronounce it, both are okay, literally translates into, to quote, 12 fingers. It forms the initial segment of the small intestine. It's roughly 25 centimeters long, and it originates, like I said, at the pyloric sphincter. It arches into a C shape as it turns or curves around the head of the pancreas, and then becomes continuous with the jejunum at the duodenojejunal flexure, shown here in this picture. Most of the duodenum is retroperitoneal, although the very proximal part is intraperitoneal and connects to the liver by the lesser omentum. Within the wall of the duodenum is the major duodenal papilla, through which bile secretions and secretions from the pancreas will enter the duodenum. There may be a minor duodenal papilla, maybe not. Not all individuals have it, which can receive a small amount of pancreatic juice through what's called an accessory pancreatic duct. The jejunum is the middle region of the small intestine, and I need to warn you, on your lab exam, if we tag the duodenum, it'll be very close to the stomach. If we tag the jejunum, it'll be somewhere in the middle of the intestines. If we tag the ileum, it'll be close to the ileocecal valve. So the duodenum, your landmark is the stomach. For the ileum, your landmark is to look for the ileocecal valve. Anything else tagged in the middle, you're going to go for jejunum as your answer choice. The jejunum is the primary region within the small intestine for chemical digestion and nutrient absorption. It is intraperitoneal and is suspended in the abdomen by the mesentery proper. Earlier I just said that the duodenum is attached to the liver. I don't want you to think that it's continuous with the liver. The tube itself is not attached to the liver. I was trying to impress upon you that it is attached to the liver because it's wrapped up in that lesser omentum. Moving on to the ileum. The ileum is the final or last region of the small intestine. It terminates at the ileocecal valve. This is a sphincter that controls the entry of material into the large intestine. Once it goes into the large intestine, that is called feces. Internally, on a macroscopic level, we can see folds in the mucosa and submucosal tunics. These are called circular folds, also referred to as plicae circulares. Their job is to slow down the passage of material undergoing digestion, allow more tumbling action of the chyme inside. This allows for proper mixing of the chemical enzymes, and it increases the surface area of the chyme to interact with these digestive enzymes. We definitely have models in the laboratory where you can see the plicae circulares. We also can see these visually in the cats when we cut open the small intestine. Histologically, the circular folds, when we view them under a microscope, we can see finger-like projections of the mucosa only. These are called villi. So in the right hand picture, you're seeing an intestinal villus. These are again extensions of the mucosa only. Plicae circularis are extensions of the mucosa and submucosa. The purpose of the villi is to further increase the surface area of the small intestine for absorption, as well as for interaction between the secreted enzymes and the chyme that is tumbling along in the lumen. To increase the surface area even more are microvilli. Now, microvilli, these are not visible with the microscope. Instead, they collectively appear as a brush border that resembles a brightly staining surface on the apical end of the simple columnar cells. Each villus contains an arteriole and a venule with a rich capillary network in them. We have a model to show this. And there are also in the villi a lacteal. And that, remember, is a vessel for lymphatic fluid. And this lacteal is also responsible for absorbing lipids and lipid-soluble vitamins, which would be too large to be absorbed by the capillary beds themselves. So in the far right picture, you can see the lacteal in between the capillary bed. Again, the microvilli are increasing the surface area. These are extensions of the cell membrane. It creates what's called the brush border of the simple columnar cells that are found in the mucosa of the small intestine. Now let's focus on intestinal crypts. Intestinal crypts and intestinal glands. Between some of the intestinal villi are invaginations of the mucosa. Now these are invaginations. These are called intestinal glands, also known as intestinal crypts or the crypts of Lieberkühn. These glands extend to the base of the mucosa, and they resemble, to be honest, the gastric tubular glands that we saw in the stomach. Lining these intestinal glands are simple columnar epithelial cells and goblet cells. And also we're going to see enteroendocrine cells. The enteroendocrine cells have names. They are I cells, S cells, and K cells. And these individual cells are acting like endocrine glands, and they will release hormones into the bloodstream. The hormones they release are called secretin, cholecystokinin, and gastric inhibitory peptide, which these days gastric inhibitory peptide is now called a much longer, horrendous name called glucose-dependent insulinotropic peptide. I will address these three hormones on the next slide. These hormones have very important roles in the process of digestion, and they deserve your attention. Going back to the intestinal glands, some histological features are the submucosal glands, particularly in the duodenum. These are also referred to as Bruno's glands. They are really important for making a viscous alkaline mucus, and that protects the duodenum from the acidic chyme that's being released from the stomach. Circular folds are best developed in the jejunum, or the plicae circularis, as I like to call them, and they're almost pretty much absent in the ileum. Additionally, villi are larger and more numerous in the jejunum. The number of goblet cells in the mucosa increases from the duodenum to the ileum due to the increased need for lubrication, as digested materials are absorbed and undigested materials are left behind. Lastly, Peyer's patches are very abundant primarily in the ileum. The Peyer's patch is represented in this picture as a lymphatic nodule. I want to remind you that a Peyer's patch is mucosa-associated lymphoid tissue, and it represents a single B cell that has come into contact with its antigen and has replicated and divided to create a colony. The intestinal glands have enteroendocrine cells, and these enteroendocrine cells have names: I cells, S cells, and K cells. and I'm going to explain to you the hormones that they release and what those hormones do. After this slide, you should definitely pay attention to the 4 total GI hormones that I have now since taught you. One was gastrin from the G cells in the stomach, particularly the pyloric glands. The other 3 hormones shown here come from intestinal glands, specifically enteroendocrine cells that are found within the intestinal glands. Special cells release these hormones. The hormones from the intestinal glands are cholecystokinin, gastric inhibitory peptide, and secretin. Gastrin, like I said before, is secreted by the G cells in the antrum of the stomach in response to stretch of the stomach and the presence of peptides or amino acids, or under the nervous stimulation from the vagus nerve or from the enteric nervous system in the submucosal plexus. Again, gastrin hormone will stimulate parietal cells to secrete hydrochloric acid. The remaining 3 hormones are secreted by intestinal glands of the small intestine, specifically found within the duodenum and or the jejunum. Secretin is secreted by the duodenum in response to acidic gastric juice emptying into the duodenum. Secretin promotes pancreatic secretion of bicarbonate mucus from the pancreas. This will help neutralize the hydrochloric acid from the stomach. It also promotes pepsin secretion from the chief cells and inhibits gastric acid secretion by parietal cells. Gastric inhibitory peptide is secreted by the mucosa of the duodenum and jejunum, mainly in response to the presence of fatty acids and amino acids. Its job is to inhibit gastric acid secretion and slows the emptying of the gastric contents into the duodenum. Gastric inhibitory peptide has since been renamed to glucose-dependent insulinotropic peptide. Its new role, or additional role, has been this hormone will actually go to the pancreas and stimulate the pancreas to release insulin. You will learn more about this GIP hormone in physiology. Cholecystokinin is secreted by the cells of the duodenum and jejunum, mainly in response to digestive products like fat and fatty acids and glycerides in the intestine. Cholecystokinin stimulates pancreatic enzyme secretion and gallbladder contraction so that bile stored in the gallbladder is released into the duodenum and can emulsify fats. Not a hormone, but still found within the intestinal glands, are duodenal glands, like I mentioned in the previous slide, also called Brunner's glands. And again, they secrete a bicarbonate mucus so that the acidic contents of the stomach do not promote digestion of the intestinal mucosa, leading to an ulcer. So that's very important that you understand those 3 hormones from the intestinal glands. In the brush border of the small intestinal cells, one can isolate several different enzymes that help in digestion. First, there are a few enzymes involved in carbohydrate digestion, such as maltase, lactase, and sucrase. These enzymes split larger sugars, like tri- and disaccharides, into monosaccharides. There is also an intestinal peptidase. A peptidase breaks down small peptides into individual amino acids. There is also an intestinal lipase that digests triglycerides into free fatty acids and monoglycerides. Lastly, and this is really important, there is an enzyme called enterokinase. The job of this enzyme is to activate pancreatic enzymes, most notably trypsinogen, into active trypsin. Once trypsin has been formed, it can then activate its cousin enzymes also secreted by the pancreas. These enzymes include chymotrypsinogen and procarboxypeptidase. I will discuss these enzymes in just a little bit. We're now finally at the large intestine, or large bowel. It forms a 3-sided perimeter in the abdominal cavity. It pretty much takes on an inverted U shape. It's called the large intestine because of its diameter, not because of its length. Remember, the small intestine is much longer, but its name is small intestine because of the small lumen that it has. On average, about 1 liter of remaining material passes from the small intestine to the large intestine daily. The large intestine's role is to absorb most of the water and ions from the remaining digested material. In so doing, the watery material that first enters the large intestine soon solidifies and becomes feces. As soon as the chyme passes through the ileocecal valve, shown here in the top right picture, we now call it feces. The large intestine will store the feces until the body, or you, the person, is ready to defecate. The large intestine also can absorb a small percentage of nutrients. Mostly, again, it's water. It can also participate in some vitamin synthesis, particularly vitamin B complexes, and even vitamin K. There are some bacteria, there's a lot of bacteria of different kinds that actually line our entire alimentary canal. Some of these bacteria can also aid in digestion. Again, main function is to absorb water and some remaining electrolytes. There is the movement that the large intestine can participate is in peristalsis, excuse me, and this idea of mass peristaltic movements. I'm just going to tell you about some of the reflexes and these movements right now. The large intestine movements are regulated by local reflexes in the autonomic nervous system. With the ingestion of more food, there are peristaltic movements in the ileum that increase, as does the frequency of the opening of the ileocecal valve. This is called a gastroileal reflex. It results in the accumulation of more chyme in the cecum and ascending colon. Actually, technically at that point, we call it feces. Segmentation movements are infrequent in the colon. Instead, 3 types of movements are typically associated with the passage of digested material through the large intestine: peristaltic movements, haustral churning, and mass movement. The peristaltic movements of the large intestine are usually weak and sluggish, but otherwise they resemble those that occur in the wall of the small intestine. Haustral churning occurs after a relaxed haustrum fills with digestive or fecal material until its distension stimulates reflex contraction in the muscularis. I'll remind you, smooth muscle, when stretched, responds with an increased force of contraction. The puckers that you see in the transverse colon, shown up here, those little pouches that you see along the large intestine, those are called haustra. So each of those little pouches can contract on the material inside. Mass movements are powerful peristaltic-like contractions involving the tiny E. coli that propel fecal material toward the rectum. Generally, mass movements, like I told you before, occur 1 to 3 times a day, often during or immediately after a meal. This is called the gastrocolic reflex. In fact, in physiology, these reflexes that I just told you about, gastroileal, gastrocolic, we call these movements of propelling the chyme and feces further down the alimentary canal the law of the gut, or as we like to say, food in, poo out. Within 20 minutes of starting to eat, these reflexes reach your large intestine and feces are moved towards the rectum. 2 common problems with defecation are constipation and diarrhea. Constipation is typically a temporary impaired ability to defecate and generally results in compacted feces that are difficult to eliminate. Constipation may result from a combination of factors including a diet low in fiber, dehydration, lack of exercise, and improper bowel habits. Not defecating when the urge arises, allowing additional water to be absorbed from the feces, is not good, and you can actually get a lazy bowel. There are some people, particularly toddlers and teenagers, they get FOS, full of, you got it. And because they fight back those mass peristaltic movements and fight back the urge to defecate, the gut stops trying, and they can get impacted with fecal material. Some children have to be hospitalized and retrained how to obey the law of the gut and how to respond when they feel that pressure in their bowel. Diarrhea may result as a consequence of disruption in normal mechanisms that absorb intestinal water. So basically, you get an excessive amount of water staying with the digested material, and this causes a very loose stool. When we look at the histology of the large intestine, we see, again, intestinal glands. These intestinal glands are much, much more rich and abundant with the goblet cells. We started to look at this histology in our first week of Lab Unit 4. Look at the right picture on this slide. All those clear-looking, almost cloud-like cells that you are seeing are the goblet cells filled with mucin. They are releasing the mucin. It mixes with water to create mucus. This helps the fecal material slide through all the sections of your large intestine. Features, some macroscopic features of the large intestine. It's divided into several sections. The initial or first region of the large intestine is a blind sac called the cecum, which is located in the right lower abdominal quadrant. This pouch extends inferiorly from the ileocecal valve, which represents the attachment of the distal end of the small intestine to the proximal region of the large intestine. Projecting from this region of the cecum is the vermiform appendix. It is a thin, hollow, finger-like sac lined with lymphocyte-filled lymphatic nodules, or another way of saying that is mucosa-associated lymphoid tissue. Both the cecum and the appendix are intraperitoneal organs. At the level of the ileocecal valve, the colon begins and forms an inverted U-shaped arch. The colon is partitioned into 4 segments: the ascending colon, transverse colon, descending colon, and sigmoid colon. Let me review this. The large intestine has A. The cecum with its vermiform appendix. Superior to the cecum begins the colon. There are 4 subsections of the colon, and these subsections together with the cecum and the appendix create the large intestine. The very last part of the large intestine is not the colon, but now is referred to the rectum. So all of the items shown here in this picture, with the exception of the anal canal, are part of the large intestine. The ascending colon originates at the ileocecal valve and extends superiorly from the superior edge of the cecum and goes upward until it bends, and this bend part is called the right colic flexure, sometimes referred to as the hepatic flexure because it's close to the liver at this point. Then we have the transverse colon, and the transverse colon is attached to the posterior body wall by the transverse mesocolon. And then we're going to see another turn. This is called the left colic flexure or splenic flexure because the spleen is found on the left side. Now we have the descending colon, and the descending colon is going to end at the sigmoid colon. It's called sigmoid because it looks like it, because of the turns it makes, as the letter S looks like it. And this sigmoid colon begins at the sigmoid flexure. Again, the bends of the letter S is what we should be thinking of. And it is attached to the posterior body wall by the sigmoid mesocolon. Finally, the large intestine ends as a retroperitoneal structure that connects to the sigmoid colon, and this is the rectum. All of the mucosae for the large intestine, by the way, consists of simple columnar epithelium. The rectum has 3 transverse folds called rectal valves, and these valves are very useful to us because they allow us to pass gas without letting go of our fecal material, which I think you would appreciate that considering how many times you pass gas on a daily basis. that you're not letting some of your feces get into your underwear while you do this. If you look at the picture on, in front of you, you're seeing pouches of the large intestine. Those are called haustra. They're puckered sacs. What makes the large intestine puckered like this? It's because of a thin band of longitudinal smooth muscle. This longitudinal band of smooth muscle is not labeled on this slide, but I will show it to you on the next slide. And that's called tini coli. And it's because of this band of smooth muscle contracts that it causes the puckering of the large intestine, much like pulling on purse strings to cause the opening of the purse to pucker. Attached to the tini coli in this picture are epiploic appendages. These are also sometimes called omental appendices, and these are fat-filled pouches of visceral peritoneum. Again, we had the tini coli. Tini, remember, IA makes the sound E, and that, or I should say, the I makes the sound E, and AE still makes the sound E. So it sounds very ridiculous, I agree. Tini coli. These are bands of longitudinal smooth muscle that are shown here in the picture. Looks like a ribbon that goes all along the length of the large intestine, shown as little yellow pouches hanging from the tini coli are omental appendices, oftentimes called epiploic appendages. These are again fat-filled omentum. The haustra are the puckers of the large intestine created by the tini coli contracting. That's also the reason why your poop, your feces, comes out bumpy. It's because it's been molded to these haustra. In the bottom right picture, you're seeing a section through the rectum. You can see the rectal valves. Again, those valves help prohibit you from releasing your feces when you expel gas. Also in this picture, you're seeing external and internal anal sphincters. I will address those on the next slide. We finally have come to the last few centimeters of the large intestine, and this is called the anal canal. The anal canal passes through an opening in the levator ani muscles of the pelvic floor, and the internal lining of the anal canal consists relatively of thin longitudinal ridges called anal columns, between which are small depressions called termed anal sinuses. As fecal material passes through the anal canal during defecation, pressure exerted on the anal sinuses causes their cells to release excess mucin. As a result, this extra mucus lubricates the anal canal during defecation. At the base of the anal canal are the internal and external anal sphincters. These close off the opening to the anal canal and relax or open during defecation. The internal anal sphincter is involuntary and consists of smooth muscle. The external anal sphincter is voluntary because it has skeletal muscle. Below the pectinate line, below the anal valves, that's called the pectinate line, you have sensory information. You have what we call somatic afferent. You are aware of what is coming out of your anus at this point. Sometimes mucosa can rip, and you will feel that when you are defecating. And also below this pectinate line, instead of simple columnar epithelium, the epithelium changes at the level of the anus to stratified squamous epithelium. Again, that's to help withstand the abrasion from the fecal material coming out of your body. That concludes segment 3 of our digestive system. In our last and final segment, our last 10 slides address the accessory glands of the digestive system.